Good afternoon, students. We are back again. Before I start this lecture, is anyone out there having any questions over my last lecture? If you have any questions, you can unmute right now and ask your questions. No. Okay, so we go ahead with today's lecture. I'll continue from where I had left off. So I think in the last lecture, I had extensively dealt with the issue of plagiarism, and I had also discussed some cases of plagiarism and shown you how exactly it looks like. And I also discussed a case of self-plagiarism. Then I also told you about how to protect your knowledge, especially when you are involved in some frontline research and you expect that you are making some path-breaking research, etc., how to protect your ideas, etc., that I had already told you last time. So we move on from that point onwards, and then today we discuss the issue of redundant publications. So what are these redundant publications, and how do they affect the scientific community as well as the publications? So as per the COPE guidelines, redundant publication, it happens when two or more papers without full cross-reference, they share the same hypothesis, the same data, discussions, results, etc., as well as the conclusions. So a rough criteria for redundant publications would be that they will have similar hypothesis, they will have similar sample size. What do you mean by similar sample size here? Basically, this refers to biological publications or research, where a sample size would mean, say, number of human beings who are being examined and things like that. And then the methodology, research methodology, which is undertaken is very identical. And the results are also very similar in nature. And you have at least one author who is common in both the manuscripts or publications. But most importantly, it's seen that there is no new information which is generated from these publications. And even if there is some new thing which is written up, it will be totally irrelevant to the context. Okay. So that these are the criteria for the basic criteria for redundant publications. So because of these things, the redundant publications, they actually put into question the integrity of scientific literature. Okay. And they also overweigh the relative importance of published findings because they are all duplicated. And finally, of course, the CV of the authors, the academic record, is rather distorted. Then, so what are these redundant publications? First, you have a duplicate publication. So this is a kind of a self-plagiarization of your own papers. And such a publication would be fairly similar to an article which has been published previously by the same author or the group of authors without any cross-linking, that is, without any cross-references. Then I have discussed one paper last lecture where you have very same similar manuscripts or papers which were published almost at the same time, and they resulted mainly because the same manuscript literally was sent to two different journals at the same time. And then another way to get a duplicate publication is if you have published several works earlier, you combine all of them and then make another publication out of it. Okay. And finally, of course, when you work on different samples, okay, which give exactly the same results. Okay. So there is no fun in working on different samples when they are all giving the same results. And in fact, this happens. I have seen this happening in many PhD theses also. So we have chapter after chapter which is showing exactly the same behavior, same curves, and very same kind of conclusions all through the thesis. So this is an example of what you mean by saying different samples giving exactly same kind of results. Then another kind of this duplicate publication or salami, or what is called salami slicing. See, salami slicing, of course, you may be wondering what it is. It is rather alien to our Indian culture. It's a European dish actually, a Western dish, where you make fine slices out of something, and then so that all slices, of course, they look same. It's a food item. So salami slicing is rather unknown to us. So I would say it's quite similar to what we would call a papadi slicing. So then what happens in this salami slicing? So here what happens is that you have one set of study, okay, one study out of which you squeeze out at least 3 to 4 publications from the same single source of study. And how will this happen? You can see that, for example, you study the behavior of some property of a material across some temperature region from, say, 5 degrees Kelvin to some 500 degrees Kelvin. Okay. Now, what you try to do is you have a brilliant idea and you say that, look, I will try to first publish the study from 5 Kelvin to, say, about 200 Kelvin. And then I'll publish the study from 200 Kelvin to room temperature. And then finally, I'll publish another paper which gives the study from room temperature to 500 Kelvin. So what you're essentially doing here is that you have one set of measurement which you have carried out and you have broken it up into 3-4 sets and then trying to get partial publications from the single source of study. So what will happen, because unless the behavior is drastically different, it will be very similar to an article which is published previously by the same author, again without cross-linking. So these are kind of problems you have with redundant publications. So basically they constitute an unethical practice. So what you are essentially doing through redundant publications is that, first of all, you are wasting the time of the editors and reviewers of the journal to which you are sending these papers. And then the CV of the author will get bloated up by all these duplicate publications. And so why do people indulge in these kind of publications? So the reasons given by many people are quite shallow. Okay. So sometimes they say we did not read the instructions. Sometimes they say that, oh, we would like to reach different audiences with these different papers. Okay. Then they say, okay, we simply forgot to make a cross reference to our earlier papers. Then some people say that, okay, we think that the overlap is much less than what the editor or reviewer thinks. Okay, as if that they know much more than the editor or reviewer. It can happen, but not always. Then again, they also very glibly say that, okay, we broke the rules, but that was not our intention. So these kind of shallow reasons are given by people when they are caught. Okay. So again, so what happens basically because of these duplicate publications? You're using up the editorial resources. See, the time required in receiving a manuscript by the editor, then assessing whether it is reviewable or not, then sending it to the reviewers, and then getting it reviewed. Then also, see, all journals have a certain limitation on number of pages. And if in case this kind of manuscript gets published, you are basically using up the number of pages which could have been otherwise used by a more useful publication. Okay. And then again, when suppose you are caught at the end of the whole thing, then you have to have a retraction and you have a public declaration that is a duplicate article. That also takes place. Then more importantly, you also deceive your readers, because the readers, when they are led into reading the journal, the manuscript for the first time, the paper for the first time, they feel that okay, it is something original, something new, etc. But then later it is realized that it is not original. And then also there's a question of ethics here, because whenever these days, whenever you submit a manuscript, you have to submit a signed declaration that these set of results or this manuscript has not been submitted elsewhere to any other journal, it has not been published earlier. So these kind of declarations will have to be signed declarations will have to be given. And then once it is of course published, then there's an issue of copyright. Okay. And so all these things are broken by this practice. Okay. And then another thing is that you have a qualitative distortion of scientific evidence, because the information is basically repeated, and there's a false perception that there is a greater scientific evidence coming out. Okay. And then there's also a quantitative distortion, because whenever you publish a paper in a journal, the journal keeps track of how many people are downloading this journal, how many people are reading it, how many people are citing this paper in their own other papers which are written later. So this is all what is called metrics or meta-analysis. So now when you do a meta-analysis on a redundant paper, it's all useless. Okay. So then finally, of course, people may get promotions and rewards based on this redundant publications also, and through which you are actually taking away the credit from someone else who might have been more eligible for that particular reward or a promotion. Okay. So to quote a person, an author in Indian Journal of Ophthalmology, it says that the foundation of scientific publication is based on trust, honesty, and credibility. Originality is a key factor in scientific progress, and if the authors cannot assure this, then one develops mistrust towards the contents of the article. The ultimate responsibility for the integrity of the publication rests on the shoulders of the authors. So, in the larger interest of mankind, these aberrations must be dealt with firmly and immediately. So that was as far as redundant publications was concerned. So now we move on to the next topic, which is selective reporting and data misrepresentation. These are also considered scientific misdemeanors, but probably not as serious as the other ones which we discussed in the last lecture. So what is this selective reporting? There's one problem with the scientific community is that whenever you conduct a scientific research, an experiment or whatever, you would like that you get all the time something good out of your research. Okay. And when I say good, basically what I mean to say is that you would like to see positive things coming out from your result. So basically it all boils down to everyone wants the good data. But the problem is whenever you do an experiment, inevitably you have data which is not so good also. Okay. But this is something which people are rather reluctant to accept, because they say that no, we don't have any bad data. This is all data and it's all good data. Okay. So you have some kind of a selective obliteration of bad data. Okay. So you just try to push it under the carpet, even though it is right in front of you. You don't wish to accept that you have good, bad data. So who doesn't like or want good data? So everyone wants good data. So even if it means that the negative or bad data is not reported at all, everyone wants to present or push only the good part of your data. So this is called a selective reporting of data, and this kind of selective reporting can be a cause for a problem in biomedical research, which is called replicability problem. Okay. So what is replicability problem? So whenever you publish a paper concerning some biomedical research, so people would like to repeat that particular problem and see whether it is true or not. So now if you have intentionally suppressed some data in your manuscript and published only a part of it, selectively you published it, so it can never be replicable. Okay. So you cannot replicate that result. So this is what is called a replicability problem. So how does this selective reporting affect? We'll see. Okay, so whenever you have certain results which you have uh, by choice, you have considered only the positive results coming out of your research and neglected the other results, which could be termed as bad, or it could be termed as negative or unexpected, whatever you can call it. Now, why does this happen? See, either this is what is called a questionable research practice, okay, or a research misconduct. But why should this happen? See, there could be some personal interest which will drive this, or it can also happen that there are certain sponsors who are sponsoring your research. They want only certain kind of data, okay. So that is how you end up by reporting positive results, which are actually a false alarm. So what will this result in? Of course, a positive result will result in a good publication, probably in a high impact journal. And once you have published in a good journal, you will get good citations. You will also get media attention. So because of all this, you may get good grants, you may get awards, you may get tenures, etc., tenure track, etc., and all those things. But then the root of the whole problem is that you are working on the basic premise of a result which is selectively presented. Okay. So let's see one example how to make, say, negative results invisible. Okay. Here you can see that there's a set of trials of a biological research here. Okay. Just one second. Yeah. Here you can see that these set of dots, they represent some 105 trials of a drug which is being tested. Okay. So out of this 105 trials of drug, actually initially there were 53 positive results and 52 purely negative results. Then what happened was the authors, they did not want to show that they had this much amount of, almost half were actually negative results. They didn't want to show that a trial on a particular drug was giving almost 50% negative result. So what they tried to do was they tried to selectively choose data which is only positive. So when the first publication turn came, it turns out that it had only 52 positive trials and only 25 negative trials, and this was published. Okay. So then once this was published, there was again an attempt which was made to even consider these set of negative results which were there and again see whether one can change the negativity of this result. Okay. And so how do you change the negativity? Of course, they were negative because of certain bad points or unfair negative points, etc. in the course of study. So suppose you neglect those bad points or neglect those unfavorable points, then that study will turn slightly positive. Okay. So once you do that, say they converted 10 negative trials into positive trials. Okay. So now you see how slowly the original set of positive results is now growing up. Okay. And the percentage of negatives is becoming very small. Okay. So now again, if you put a spin on this, the remaining negative results, saying that, oh, this could have been this way, that could have been that way, or something like that, you put some spin on that and then convert those into say neutral results, okay, something like that. So then you can see finally how few negative results are left. And in the final point, what you try to do is increase the positivity of the positive results. So make them even more positive. So they look much bigger than the negative results, which have become very small. So this is how actually you can magnify a set of positive results and then brush away all those negative or bad results under the carpet. Okay, so then we come to the next title, which is data misinterpretation. This is a very tricky thing. What happens is here is that the data which you have collected is actually real data. Okay, you have actually collected real data, but then like what you have done earlier in the positivity and negativity issue, you try to present this data, the whole of the data, in a very deceptive manner. See, this is not a hardcore scientific misconduct. In the very beginning of my lecture series, I showed you one figure where you had a perfectly ethical conduct in research, and then you had on the other side the black area, which I considered is a very bad misconduct, bad research. And there were certain areas in the middle which I called them as gray areas. Okay, so this data misinterpretation actually falls in the gray areas of misconduct. Okay, and they are not considered as severe scientific misconduct, but then they again fail to convey an honest interpretation of the data and then create a false impression to the reader. So how does it happen? It makes one's results, which are appear more significant or convincing than they really are, and they draw certain unwarranted inferences from the presentation. And sometimes you can use even language, suggestive language, with some rhetorical effect. Let's see how this works. Okay, so before that, so the common types of misuse of statistics. Okay, so you can, for example, in a time period data, you strategically pick only that time period which is suitable for you, or you can truncate an axis artificially and then show some data, or you use a percentage change in combination with a very small sample size. Okay, then you can have this purposeful and selective bias in reporting, or you can do certain data fishing or misleading data visualization, and such a few more like that. Okay, let's see how it works. Now I'm showing you some data. This is my own data actually. Okay, this is, I have measured this data myself. So this data basically here it shows the full width at half maximum of some diffraction pattern. What is a diffraction pattern? This is a typical diffraction pattern. Okay, so this is a neutron diffraction pattern, and this is how a diffraction pattern looks like. You have a collection of peaks, and then what I do is to look at the resolution of the instrument across this 2 theta or the scattering angle. I measure the full width at half maximum of each of these peaks. Okay, and then I measure the full width of half maximum and plot it as a function of this 2 theta. So that is what is called a resolution curve. Okay, so I plot it like this and I get a curve like this. So this curve ranges from say 0.275 0.27 or 28 or something like that, and goes to about say 0.37 or something like that. Okay, so this is the actual data. But suppose now the text which is actually describing this data, suppose in my the text I write that I don't see much variation in my data, then obviously I cannot show my data like this. So I have to show my data as if that it is not varying much, at least visually, with the x-axis. So what do I try to do? I simply increase my y-axis. Okay, instead of ending at 0.38 or something, I end it at 0.5. So immediately this gets suppressed and looks flatter. And I want to make it even more flat. I don't even show numbers here and then put some very high number here so that this gets totally suppressed, and then visually it appears as if there is not much change across the scattering angle. Okay, so this is like what is called fooling with the axis, and you try to show the data in a different light depending on how you want to describe that set of data. So in reality, this data and this data are same, but only difference here is that the y-axis for the two curves is different. And here I have shown you selectively that without mentioning what is this point here, that I say that simply I write in the text that I don't see much variation across the scattering angle. Okay, that full width at half maximum is almost invariant uh, uh, with 2 theta or the scattering angle. See, basically, see what happens here is that you have to look at the scientific content here. Okay, what is it actually describing? Okay, so even if such a curve is shown, but with proper language and scientific argument, this also is acceptable. Okay, so but then you don't want to show that way, and visually you want to make it look as if it looks flat. So this is how one can try to fool with the axis and show misrepresent the data. Another example is that this global warming curve, and it shows that this first curve, in fact, it shows from this point to this point that the global air temperature change actually has come down as a function of time. But the problem is here it is showing data only from 1998 to 2012. And this was actually a part of another large curve, which was not shown in this section. So now if you see slightly differently from 1980 onwards, now you can see that it is actually a rising curve from this point to this point. And even if you actually look even back and go from even earlier times, now you can see the whole change from across the last 100 years. So this shows that actually global warming is a reality. So there are people who say that global warming is all a hoax and there is no such thing as a global warming. So those kind of people, they show only those set of curves which suits their argument. Okay. But the reality is this, where global warming is a reality and it is increasing with time. This is another something which is used very often by companies. Okay, so a person who looks at this first, he will just read this value, some KFC something, calories is 650. So this is KFC, it is showing 650, and its competitors are showing twice the value. Okay, so you feel that, oh, this is 650 and this could be 1300. But in reality, if you look closely here, the x-axis starts at 590 and not 0. Okay, so it's a misleading advertisement which indicates that this product here, it contains half the amount of calories as the other 3 products. Okay, but in reality, the x-axis is not starting from 0, it has started from somewhere higher up there. So naturally, if you do that, it will show something like that. And people who don't look at this bottom line, they will feel that visually, oh, this is half the value of this. So that is how you can actually use the manipulation of the axis and the coordinates and then show data in a different light. Okay, so now coming to the question, can we really prevent scientific misconduct? Okay, so we have now seen that this misconduct in research, it varies from unintentional errors, which I would term as honest errors. And then on the other hand, we have outright fraud and blatant misconduct. But it varies from this point to that point in a very continuous manner. There is no hard division between the two. Okay. And because of this, it is necessary that institutions involved in scientific research and the managers of scientific research, they should take more responsibility to ensure ethical and sound research environments in the country and also avoid even minor breaches of good scientific conduct. Then again, the research community as a whole must take responsibility even for people who are actually into scientific misconduct. And there must be regular seminars and courses which must be organized at all institutions on why this scientific misconduct happens, what happens when such a thing is undertaken, and what could be the consequences of scientific misconduct. All these points must be made aware to the younger generation of people for their own sake. Then, of course, I have shown that the big three of scientific misconduct, which is fabrication, falsification, and plagiarization, they should have more strict definitions so that legal measures can be taken against the perpetrators of these scientific misconducts. And for people who do what I said, the lesser misdemeanors, like what I just now told you, like data misrepresentation or selective reporting, there could be some relaxation for that kind of misconduct. Okay. And then these guidelines and regulations must be simplified and made readily available to all researchers. And then, as I said, this training, research training must include ethical and legal issues. And young people should be informed about, made aware of these issues effectively. Then, whenever serious scientific misconduct events or incidents happen, okay, there must be some kind of mechanisms which are independent and very effective, which can be taken up at a national level in all countries. Of course, we are now bothered mainly about our own country. So I think it is high time that our own institutions take more interest on this kind of a mechanisms which can investigate these incidents of misconduct. Then finally, of course, one should reduce emphasis on this number of publications. Okay. See, that has given rise to a lot of problems, a lot of issues and misconduct. So if only this emphasis on publish or perish kind of an attitude is reduced, it can actually definitely result in a more healthy culture of transparency and proper ethics could be established. Okay, now we move on to the next issue, which is called ethical issues and ethical committees. Now, these are something which are mostly related to the medical and biomedical research which takes place, and these things have like ethical committees, etc., are a result of scientific misconduct which is being conducted or carried out in these areas of research. So, if you consider biomedical research and ethics, first, who are the people involved here? So, we have medical doctors and researchers, then we have biomedical researchers, then we have pharmaceutical companies, and then whenever you have this pharmaceutical company making drugs, etc., there is what is called a connection between medical practitioners and the pharmaceutical companies. Let's see how they are involved here. So when you consider a medical doctor's primary obligation would be concerning his patient's health and well-being, and he should be mainly interested in improving the quality of life. But what happens when the same person actually dons the role of an investigator in research, say for example in trial of a new drug or a new drug program, then there is an inherent conflict of interest. I'll tell you more about what is this conflict of interest in the coming slides. Now, why does this happen? This is because the doctor would be required to recruit the patients for a trial, and he has to also generate some good data, okay, for the study, and also perform certain non-therapeutic procedures which are called randomization of trials, administration of placebos, and some trial protocols, etc. So, but then while doing all this, he may actually neglect taking care of his patient, and because of which the patient's health may actually deteriorate. So, is it possible for a doctor to fulfill the obligation of medical care on one hand and at the same time conduct these clinical trials? So, now we have something called the Helsinki Declaration. I'll talk about this in more detail. So this declaration, it says that it is the duty of the physician to promote and safeguard the health of patients, including those who are involved in medical research. So the physician's knowledge and conscience are dedicated to the fulfillment of this duty. So now we come to the biomedical researchers. So these people in general, they are not actually practicing doctors or physicians. They are people who are actually involved in biomedical research. They develop new drugs. These people develop new vaccines for the society, etc. But then when you develop a new drug, it is not as if that you sit in a laboratory, you make a new drug and immediately use the drug on the society. That is not possible at all, because making of a new drug involves a lot of steps. It involves a lot of clinical trials, and these trials actually involve participation of human subjects, and because of this they cannot escape ethical issues. And also these people, so if you now look at, say, for example, how much time it takes from, say, research into a new drug and final implementation of the drug or a vaccine. Okay, let's see a typical, this is the FDA of United States. So you can start with basic science research. It takes 2 to 5 years. Then we have what is called preclinical testing, 1 to 2 years. Then we have clinical trials. See, preclinical testing would be testing it in the laboratory and on animals, etc. Before you start moving on to clinical trials on humans, and that takes almost 9 years. And how is it? This split into testing in healthy subjects, testing in individuals with disease, then larger scale testing in individuals with disease, etc. Then finally, after this, you have the approval stage. It takes almost more than 6 months to 2 years sometimes. And finally, you end up with an approved drug which is released in the market. So all this process, imagine a company, maybe a private company who is developing a drug, it takes almost 10 years to make this drug come into the market. So imagine now how much money this company has to spend in these 10 years in the development of the drug. So there are huge amounts of money involved and things like that. Okay. So, so when such a thing is concerned, naturally there will be certain issues, ethical issues which come into play here. Okay. So let's go to the next point. Let's see how these pharmaceutical companies work. So in a capitalist scenario, basically, most of the world is capitalist. You can see the goal of any industry is to make money. Okay, as much money as possible from their whatever they invested. So for most industries, as I said in the beginning, business ethics is an oxymoron. There is no such thing as business ethics. They want to make money from whatever investment they have made. But then the issue is, if it's a steel industry, it's fine. Okay. Or if it is some other carpentry industry, it's fine. But then when you talk about pharmaceutical companies, the situation is quite different, because these companies are manufacturing and selling drugs to improve the health of the society. Okay. So in that way, this particular industry is quite unique. And because of this, it faces a lot of ethical issues, which are very unique to this industry. So on one hand, can profit making be most important concern for the company? Okay, if it prevents people from purchasing, say, life-saving drug, because if life-saving drug is so expensive, not many people can buy it. Okay, so the company is actually stopping people from buying the more people from buying the life-saving drugs. Or should these companies be allowed to put a price on human life by charging exorbitant price for these drugs? And do these companies have a moral responsibility to supply affordable drugs and to invest in developing new treatments? So these are all issues which these pharmaceutical companies face, and these are all ethical issues. Okay. And then finally, we have a connection between the medical doctors and these pharmaceutical companies, which probably most of you are quite aware of. Because, as I said, a medical doctor is supposed to extend medical care and advice to his patient, and also, not only that, prescribe the right drugs which are available at reasonable prices. Now, a pharma company which is actually making drugs, it always tries to maximize its profit from the sale of drugs. So what it does is it induces the doctors to prescribe the drugs made by them to their patients. So this is the problem which can create conflict of interest and other ethical issues. So when a medical doctor actually participates in a program which is sponsored by a pharmaceutical company, there's a distinct possibility of his professional autonomy getting compromised. Okay. Or you can very often see doctors prescribing certain drugs to of a certain company to their patients because they have received some undue financial inducement. So, because of this, the Medical Council of India it stresses on the fact that in dealing with pharmaceutical and allied healthcare industry, a medical doctor shall always ensure that there shall never be any compromise either with his or her own professional autonomy and or with the autonomy and freedom of the medical institution there. Okay, so what are these ethical committees? In our country, of course, I told you about FDA in the United States. In our country, we have something called CDSO, which is a Central Drug Organization. And every institute, research institute organization dealing with human beings as subjects of research is required to form an ethics committee, which has to be registered and must follow the guidelines issued by the CDSCO. And this ethics committee has to comprise medical, scientific, non-medical, and non-scientific members. And what is the responsibility of this committee? It is to ensure the protection of the rights, safety, and well-being of human subjects involved in a clinical trial. And then it shall be responsible for reviewing and approving the protocol, suitability of the investigators, facilities, methods, and many other such things. Even our organization DAE, all the units have wherever they are working with biomedical research topics, they have an ethics committee. BRC has an ethics committee in this regard. Okay. So this ethics committee must allow inspectors who are authorized by the government to enter its premises, inspect all the records, etc., related to the trials, clinical trials, and then give proper replies to all the queries which are raised by these inspectors. And if the ethics committee fails to comply with any of these conditions, then the licensing authority may actually withdraw the license or the permission accorded to the institution to conduct the particular medical trial or something. It is called the Central Drug Standards Control Organization, Government of India. So why did this all come about? Okay, I'll just give you 2-3 small stories here. See, what happened was during the Second World War, the Nazi medical doctors, the German medical doctors, they conducted certain inhuman experiments on the prisoners in the concentration camps. So what were these concentration camps? Because the Germans at that time, during the time of Hitler, they believed that the Jews were to be exterminated from the world, and they rounded up all the Jews and then sent them to concentration camps where they were treated in a very inhuman manner. So these prisoners, including children, were actually forced into participating in experiments that were conducted without anesthesia and resulted very often in death, trauma, disfigurement, or permanent disability. And all these are considered now as examples of medical torture. And of course, after the war, these doctors were tried for their crimes at the trials which were conducted at a place called Nuremberg in 1947, which led to the establishment of the first code of medical ethics called the Nuremberg Code. And this was actually called the doctor's trial, in which some doctors were sentenced to death and executed, and some were actually imprisoned. So this was the actual trial which was going on in 1947 at Nuremberg. So the Nuremberg Code laid down certain guidelines. First, it said you must have voluntary informed consent. That means the human being must actually agree to the test which is being conducted on him. So the consent has to be voluntary. Okay. And also, of course, he should have the legal capacity to give that consent. Okay. And then, of course, it shows that it should have sufficient knowledge and comprehension of the elements of the subject matter involved so as to enable him to make an understanding and enlightened decision. Okay. So before acceptance on affirmative decision, they should be made known to him the nature, duration, and purpose of the experiment, and the method and means by which it will be conducted, all the hazards which are expected, and the possible effects on his health or person. And then it is the duty of the responsibility for ascertaining the quality of the consent. It rests upon each individual who initiates, directs, and engages in the experiment. And of course, this person has to directly deal with it, and it cannot delegate it to any other person. Then fruitful result for the good of society. So the experiment which is conducted should be such that it must yield some fruitful results for the overall good of the society. Then prior experimentation on animals and knowledge of the problem should be already present. So that means whatever trials you are going to do on humans must be first done on animals, what is called animal experimentation, and then based on the results which come out of that, only then you go ahead with the human experiments. Then you must try to avoid any unnecessary physical or mental injury to the human subjects. Okay, so whatever experiments are conducted, they must avoid any kind of physical or mental injuries. Then any non-lethal or disable procedures must be not used at all. Okay, so no experiment can be conducted where there is an a priori reason to believe that it could result in the death of the human subject. Then the degree of the risk should never exceed the benefits. So this is a balance which has to be always kept in mind, that the benefit balance has to be much higher, better than the degree of risk. Then proper preparation and facilities to prevent injury or death. Then performance of the experiment only by scientifically qualified people, not anyone else. Okay. Then participants may freely end the experiment. So in case the participant feels that he wants to put an end to the experiment on himself, it should be possible to for him to get out. And then if it's found that the experiments are too dangerous, they should be immediately, the whole project must be stopped. So this was the first code of conduct, which was called the Nuremberg Code. And following that, of course, there were many other declarations which came out. The next one was Declaration of Geneva in 1948. And these declarations, they are pertaining to certain aspects of these trials and the medical profession. And they keep getting updated also. Then there is what is called the Declaration of Helsinki, initially in 1964, on human experimentation. And that also keeps getting updated every few years. And then there was a Declaration of Tokyo, which was concerning human torture. So in this connection, I'll describe one experiment which was conducted in the United States. It was called a Tuskegee experiment. It was called the Tuskegee syphilis study. It was an official American governmental research project from 1932 to 1972. And what was this project? It was basically a totally unethical experiment on Afro-American patients who were mostly agricultural, rural laborers. So there was a group of about 400 infected patients and 200 uninfected control patients who were recruited. And then they were actually examining the natural course of untreated syphilis. Okay, that means they wanted to see how this disease called syphilis, if it is untreated, would finally result. In what will it result in? But then unfortunately, the subjects were not aware of the actual nature of the project. And they were all told that they were actually receiving treatment for bad blood. Okay. But in reality, they were not given any treatment at all. And then what happened in 1942, actually, the drug penicillin was discovered, and penicillin was very effective in treatment of syphilis. But in spite of that, this drug was not administered to the infected patients in this project actually. And the most bad thing about this experiment was that this study went on till 1972. That's hardly about 50 years from back from now. 1972, America was already, United States was an advanced country of the world. Even then, this study was going on, and it was stopped only because the story got leaked by the press to the general public that such a kind of experiment was going on. It was a totally inhuman experiment which was being carried out under the full knowledge and directions of the American government. So finally, in 1997, actually, the President of the United States issued an apology, which is very hard to come by. Any apology from the United States, it almost never happens. But in this case, it happened. And then it was frankly told that the government did something that was wrong, deeply, profoundly, morally wrong. And it was an outrage to our commitment to the integrity and equality of all our citizens. Okay. And then it goes on where finally he says that American people are sorry for the loss, for the years of hurt. You did nothing wrong, but you were grievously wrong. I apologize, and I am sorry that this apology has been so long in coming. Okay. Then finally there was another experiment which was conducted by the Japanese army. This was between 1937 and 1945. The Imperial Japanese Army, they undertook a very covert biological and chemical warfare research and development program, and they conducted these experiments on human prisoners in an occupied city in, at that time, Japan occupied part of China, and they did this in a city called Harbin in China. And this program was also supported by the then Japanese government, and they operated biological weapon production, testing, deployment, and storage facilities. And this research was conducted because they wanted to save their own soldiers by learning about how these chemical weapons or biological weapons would affect the human body, and how it would help the soldiers to prevent getting affected by the biological weapons. So they were actually tested in the field on cities and towns in China, and the estimates are really not known, but they expect that this Unit 71 estimate of people killed is almost about 500,000 people. Okay. So then what happened after the end of the war? These researchers were captured by the Russians and they were prosecuted. But then some of the researchers were actually captured by Americans. But then what Americans did was something different. They were actually given secret immunity in exchange for the data which they had conducted on human beings. Because why Americans wanted to utilize this data for their own biological warfare program. Okay, so you see the ethics part here, how unethical way the Americans behaved here. So you can see that war actually makes very strange bedfellows. Okay, so what happens is that you can see that whenever this evil is there, there was a very famous quote by a person called Hannah Arendt. This person was a Jew who escaped from Hitler's Germany at that time. So she wrote at the trial of this person, Adolf Eichmann. This is Adolf Eichmann, who was caught, and he was responsible for the death of several Jewish people in Germany. So she wrote, "Evil becomes banal." So banality of evil. What is banal? Banal means it's ordinary. So evil becomes banal when it acquires an unthinking and systematic character. It becomes banal when ordinary people participate in it, build distance from it, and justify it in countless ways. Okay. So it becomes essential. Evil does not look like evil. It becomes faceless. So this was something she wrote at that time. So she found that this person, Eichmann, who sent thousands of Jews to their death, okay, he was a very ordinary and bland bureaucrat, and he was neither sadistic, he was not a perverted, or he was not a cruel looking person. He was a very normal, simple person. And the very reason he did this because he believed that his career would advance if he followed the orders of the government at that time, the Nazi bureaucracy. So he was not an immoral monster. Instead, he performed his evil deeds without evil intentions. That is basically, if it was connected to his thoughtlessness, a disengagement from reality of his evil acts. Okay, so now you can see how even ordinary people can end up doing unethical things when they are actually when they believe that it either leads to their advancement of their career or something like that. So they distance themselves from the after effect of that unethical action and then go on by doing this unethical action. Then there is also something called ethics and environment, and that also has been put up in the syllabus here. That's why I have to put this slide. So there are environmental ethics also where ethical issues are involved when you deal with environment. Like for example, you consider say resource consumption patterns. See, you find that people in certain parts of the world they get access to more food, and there are people in other parts of the world who do not have access to the food. So if you look at the consumption patterns of food, there's a large inequality here. So there is a need for equitable utilization of food resources. Then the north and south part of the globe, you can see that there is a disparity in equity. And basically you have the developed world and the third world or the undeveloped world. Then you have what are called the urban and rural equity issues. Then the need for gender equity is also there. Then preserving of resources for future generations. This is an important thing because people, if you see that, for example, forests are being cleared without any thought for the environmental or climate issues. And the problem is at this point of time, some set of people can get advantage by clearing certain forests, but then what will happen to the future generations? They are not bothered. Then you have rights of animals. Okay, this is also an issue where it is found that animals have to also be treated ethically and in the proper manner. And then you have also the issue of environment education and awareness and how ethics can be introduced in these two subjects. And finally, of course, we have conservation ethics and traditional value systems of the country. And there is a intense need for incorporating these value systems in the country now because people have lost track of all these things, and it is necessary that conservation ethics and value systems must be taught to the future generation, younger generations. Okay, so I'll just briefly introduce you to the next part before ending this lecture. Now, so far what I have talked to you was about the research part, how you have conducted the research and what are the problems in conducting the research, the misconducts which happen in conducting research, and what are the ethical problems and how one can try to solve and all those things. But now I'll try to focus on a different aspect of ethics, which is called publication ethics. That means now whatever research you have carried out is now over, and now you step into the realm of the publication aspects. So when you consider publication, there are several other things which you have to worry about. So the first question you need to ask is why should you publish? Okay, so if you look at the research life cycle, you start by planning your research, then you start collecting your data and whatever field work you do and capture data. Then after you collect your data, you do certain processing of the data so collected so that it can be put into a form that can be analyzed. And then you analyze your data and then you get some results which you feel that now it's in a position to be shared to the larger public. So at this point, you come to this section where you have want to publish your results and share it to the larger part of the community. So as soon as you finish your research problem, you would like to disseminate this knowledge which you have gained. And this dissemination can happen in form of a publication in a journal, or it can be a conference presentation, or it could be a closed meeting in front of certain peer groups, etc. Then another reason, of course, you need to publish is to get recognition for your own research, and it can help you in getting your tenure or positions and also promotions in your career. Then also it is important to enlighten the peer groups associated with your own research work and what are the funding agencies in that area of research, and of course the general public to whom you owe a lot, because the general public actually pays for most of the research which is carried out in our country. Then another aspect you do by publishing is that you can fill certain gaps in the existing knowledge with your results, and then possibly identify certain future thrust areas for research. Then you can also have, as I said earlier in my earlier lecture, that you can have certain out of the box idea or a path-breaking idea, and unless of course you push these ideas forward and publish them, I mean, you will be able to direct future research in those areas also. So these are the basic reasons that you need to publish. So, okay, I'll end with this slide. I'll just mention that there is something which is called publication ethics also. Okay. So what are these publication ethics? So publication ethics exist basically to ensure that the publications, scientific publications, remain at a very high quality, and also whatever you publish, the findings that you publish will have some effect on the public and help to gain their trust in what you're doing. Okay. And then, of course, people who do some good work should receive credit for their ideas. Okay. So whenever a research work gets disseminated, either in form of a publication or a presentation before a peer group, it has to first get validated by the concerned peer group. See, immediately as soon as you publish your research work, it is not going to be effective immediately. So that research work has to be accepted by the larger community in that area of research, and who will validate that research and say that yes, what you have done is something very useful or great or whatever. Okay, and it will have the potential to shape future research or contribute to society. So therefore, it becomes the responsibility of the scientist to follow ethical practices in the research life cycle leading to the stage of publication. Okay, I would like to stop here because the time is over now. And so let me ask if anyone has any questions at this stage. You can please unmute yourself and then ask any questions or write your questions in the chat box. Any questions, please? YouTube also, no questions? Okay, there seem to be no questions so far. So I think I take it that my lecture has been very well understood by everybody. And anyway, in case you have questions, you are free to ask me next time or send email to me, and I can answer your questions. Okay, thank you very much. So see you in next lecture.